Yeah, so quick introduction to me, I guess, before I get into what I do. But I'm a physiologist, ecologist, and work on biomechanics as well, sort of how organisms are structured, how they move and resist stress and so forth. And so my general sort of interest is in how organisms deal with their environment. And in my case, I'm focused on intertidal habitats, so places where the tide is going high and low twice a day, and so all of these ocean organisms are coming out of the water part of the day. They're sitting in the sun and they're heating up, or in the middle of the night they're cooling off, and they're sort of having to deal with terrestrial conditions for part of their life. And then the water comes back, the tide comes up, and they sort of go back to maybe normal conditions, and they get a chance to feed and to breathe and to mate and so forth. So I'm sort of interested in that relatively extreme change that happens twice a day in most cases for a lot of these organisms. And so I've worked up and down the west coast of the U.S. I came to San Diego back in 2018, and so I started to sort of center my research projects down in the San Diego area. But I've sort of, for a lot of my career, focused on rocky intertidal habitats. So those places where you see things like mussels and barnacles growing on the rocks, and all of the little mobile critters that live there, the snails, the limpets, the crabs, and so forth. Again, are coming out of the water part of the day, sitting there and potentially baking in the sun, reaching body temperatures up towards ours, when normally they spend most of their day at high tide at ocean temperature, whatever ocean temperature might be, in the 50s, the 60s in Fahrenheit. And in the ocean, and then suddenly they're at 90 or 100 degrees Fahrenheit if they've been baking in the sun for an hour or two. So I'm going to talk today a little bit about work done up in Central California in the Monterey area, which is where this picture's from, and then the latter half of the talk will all be about stuff we're doing locally in Southern California. And so the general sort of questions, if I had to put them in a simple way, is just sort of how much variation in stress do organisms experience over small spatial scales. So you might imagine if we go from Alaska to Central California to Southern California, you can imagine that, yeah, there's probably differences in terms of the temperature stress that those organisms might experience. And it can be the same species of mussels living all up and down the west coast. And so those Alaskan mussels experience a very different set of weather and ocean conditions compared to their Central California relatives and their Southern California relatives. So those huge latitudinal scales, we obviously sort of intuitively expect that there ought to be differences that these individuals are feeling. But also at the scale of an individual mussel bed here, where we've got a couple of hundred mussels all sort of packed together, how much variation is there just within that bed over the scale of centimeters here? So these animals during low tide are all clammed up. They're closed up, they've got their shells closed tight, and they're essentially isolating themselves from the surrounding environment. They are being hit by the sun, of course, they're being dried off, but while they're closed up, they can't really do any regular breathing, they can't do any feeding as well. When the tide comes back, that's when they actually open those shells and they start pumping seawater through. And so in this case, this California mussel is, over the course of a couple of minutes in time lapse here, opening its shells, starting to pump that water. In the process, it can capture food, it can also do its respiration, it can breathe, it can get oxygen out of the water and offload CO2 and any sort of metabolic waste that is produced as well. So you can pee into the water and get rid of that stuff. So high tide is the opportunity to do all this stuff, that sort of vital stuff to living and breathing. And then at low tide, they sort of, again, clam up. Now, in my lab group and myself personally, we're sort of interested in how much variation we see, again, over small scales, in terms of how the individual animals are responding to those environmental changes over the course of minutes and hours and days. So what we do is oftentimes gluing things to the outside of these animals and measuring what those sensors are telling us. So on the left here, I've got, again, a normal California mussel, which you'll find down on the seashore. It's got two wires glued to it, to the shell. And those, I'll talk about what those are measuring in a second. But we run those wires back to a little waterproof case, and we can sit there and we can log data off of an individual mussel over the course of hours, over the course of days, over the course of weeks. The local microenvironment might be triggering physiology. So in terms of what we measure, we've attached lots of different sensors to these animals, but for starters here, I'll talk about measuring their internal temperature. So we actually will drill a little hole in the side of the shell and insert a little thermocouple, a little temperature sensing device in there, sort of poke the animal inside through a hole in its shell and get a measure of its internal body temperature. The cartoon cable up on the upper right there is a magnetic sensor. And so because these are mussels, kind of like clams and oysters and so forth, they have two halves of the shell, and they got to open them in order to do that breathing and feeding. We can put a magnetic sensor glued to one half of the shell. We can put a magnet on the other half of the shell, and then when those shells move apart, we actually see a change in the voltage that we're recording in that sensor. So we can tell when the animal is clammed up tight, and when it's barely opened, and when it's fully opened, and so forth. And we can record that over long periods of time. And then down in the lower portion of our diagram here, I've got what is representing a three-dimensional accelerometer and magnetometer. So in your smartphone, you've got one of these, and it tells which way the phone is oriented. So we go from portrait to landscape, it knows to flip the picture for you. Same sort of thing here. We can, from that, figure out what orientation the animal is taking in the environment. Literally, is it pointing north? Is it pointing south? Is it standing up? Is it falling over? That sort of thing. Now, mussels typically are attaching themselves to the rock, and they don't get up and run around. They're sessile animals. That's how we get away with hanging all these wires off of them and sort of anchoring them down. So you might imagine this three-dimensional orientation is not necessarily telling us all kinds of exciting things, like if we put one of these same sensors on a bird that got up and flew away, and you can see all sorts of neat stuff there. Here, what we're really sort of looking for is, are these animals moving over the course of days and weeks and perhaps following the sun or anything else like that, or are they being shoved around by their neighbors? So we put those kinds of sensors on these animals and then build ourselves a little fake mussel bed. I mean, it's fake in the sense that it's artificial in the sense that we've constructed it, but it's made entirely of live mussels, and a subset of those mussels have these sensors on them. So in this picture here, we've got data loggers in these waterproof boxes, wires coming out of them to the various mussels, and scattered in here are 12 of those mussels that have little sensors on them. And then we've packed in a bunch of other mussels, live mussels, to sort of try to recreate that dense mussel bed condition. Then we take all of this on this plastic plate, walk it out into the intertidal, bolt it down, and put it in the mussel zone, and from there we can start recording sort of what's happening over the course of a couple of weeks. So as I said, we were doing this up in this part of it, up in Central California. So this is an aerial view of Cabrillo Point. There's a lot of Cabrillo Points in California, right? This is one of them. This is right next to the Monterey Bay Aquarium, just to the west of the Monterey Bay Aquarium. And we put, in this case, two of these plates out at two different heights on the shore. So we have one in the foreground here, which is sort of the upper end of where the mussels live in these shores. And then the one in the back where I'm sitting is a little bit lower on the shoreline, at the bottom end of the mussel zone. And so these mussels, they tend to be restricted to a certain vertical range within the intertidal zone that seems to, of course, be set by where the tide will go, but also things like where they get eaten. If they try to go down deeper towards the subtidal zone, they tend to get eaten by things like sea stars, and so they tend to be restricted to this middle zone. So we sort of bookended the two ends of the mussel zone here and looked at what these animals were doing in those conditions. So what I'm going to show you here are some relatively simple graphs, some basically animations of what we were seeing. And first we're going to look at what the animals were doing in terms of when they were opening and closing. So that is gaping the shells. And so what I've got here is two graphs in the upper left and upper right, and then this is going to be a little time-lapse movie shot of, in the foreground, the high shore mussels, in the background, you won't be able to see it, the low shore mussels. And we're going to start out in this video in the early morning when the tide is low and the sun is just coming up. We're going to progress through a low tide, a high tide, and then back to a low tide. And so as I hit play here, there's going to be this group of colored dots. Each different color represents an individual mussel. And so values that are low on these graphs mean the animals are closed up tight. They're not open very well. As you see those values go up, it means the animals are opening up. So we're going to start out at low tide. Everybody's closed, basically, right? You're going to see low values. As we go through the early morning, we're seeing also the tide height changing along the x-axis here. And eventually the tide's going to come in and start washing those ones at the upper right there. So you start seeing them opening up, those dots going higher on our scale here, because they're being washed. Now finally the tide is high enough to wash our high shore animals, and they start opening up. But notice not quite as wide as our low shore animals, because they're not sort of fully submerged for minutes at a time. They're just barely getting splashed. So they're taking the opportunity to sort of open up to a little bit of breathing, but probably not a whole lot of feeding. Whereas the low shore animals had some time to actually feed as well. The tide goes back low towards sunset, and you see everybody sort of come back down to be closed as we go into nighttime. And so, to recap sort of the pattern there for the entire day, you see a lot more variation in those low shore animals that were spending more time in the water. They have no opportunity to open up wide. They have an opportunity for a couple of hours to sit there and do some feeding and keep themselves healthy. And what we see then, if you go out and just sort of look at mussels at the low end of the range and the high end of the range, is you can sort of visually notice that there's a lot of weensy, sad-looking mussels up at the top end of the range, and there's some big fat mussels down at the low end of the range. And that is almost certainly just a product of the fact that those low shore animals have a lot more of the day to spend feeding, and also they're not necessarily spending as much time out in the sun getting stressed. So let's talk about stress. Skip over this. Talk about stress in terms of temperature. So here I'm going to show you on the next slide the same sort of, actually the same day, the same time-lapse image of the tide going low, then high, then low, but we're going to plot temperature instead of whether those shells are open or closed. So again, high shore animals are graphed on the left, low shore animals graphed on the right, and here now the y-axis is temperature. So everybody is going to start out relatively cool because we're just coming out of sunrise. It's been cool overnight, but they've been out right now in the sun. You can see those high side animals getting up towards 30 degrees Celsius, so up into the 80s, up into the 90s in terms of body temperature. The low side animals, you see they got hot for a few hours or so, and then the tide came up and they went down to ocean temperature. The high side animals, it was much later in the day that they get cooled off. But we still see some persistent difference in terms of temperature here, just over the course of that one little plate. Basically, differences between whether you're on the seaward end of the plate or the shoreward side of the plate, and how much splash you're getting. As we go into nighttime, everybody's sort of cooling back down, even though they're out of the water, and so they go into a cold, low tide overnight. So we saw peak temperatures up at 35 degrees Celsius for some of those high shore animals, even though other ones were a couple of degrees cooler. And those temperature differences, both high to low shore, but also within individual groups, are pretty persistent across days. So I'll just point out over here on the far right our low shore animals and our high shore animals, and this is just sort of average daily maximum temperature that each individual hit. So our low shore animals didn't get as hot as our high shore animals, our red dots, but also the spread in temperature, daily maximum temperature for individuals on the high shore tends to get a little bit broader. They're hitting, some of these individuals are getting really hot every day, some of them are staying relatively cool every day, even though they are literally centimeters apart on these plates. And just a visual representation of that, here are two animals that had the sensors on them on this plate. They are literally about that far apart in real life. One of them is hitting 37 degrees on a particular day. Its neighbor down here is on the same exact set of conditions, the same day, is only hitting 23 degrees Celsius. So, 15 almost degree Celsius difference in terms of maximum temperature, just by virtue of where in this bed they happen to be, and whether they're on the seaward side or the slightly less seaward side of this bed, but also what their neighbors are doing, shading them or not. Yeah.
Quick question: these critters have a chance to move?
So they don't really. So in theory, they could release their little anchoring threads that they use to hold themselves down. They do have a foot that they can stick out and sort of move around, kind of like a snail. Okay. But you might imagine that in a wave-swept environment like this, that's a really risky thing to do if you're not a good, if you don't have a good hold on the ground.
Right. And are they the same species, both low and high?
Yes, they are. Yeah, so they're the same species. They when they settle here, they'll actually wash in as larvae from the ocean and find a piece of rock and settle down onto it. So it's sort of a lottery as to whether they happen to land in the low shore, the mid shore, the high shore. But from that point on, they're generally stuck more or less in that spot where they landed. So we see big differences just over very small scales. These are differences in terms of temperature that are as big as we would see if we went and sampled mussels in Monterey versus in San Diego on the same day. It might be cool and foggy up in Monterey, it might be hot and sunny in San Diego, and you might see 50-degree differences. But just looking over very small scales, you will see the same range of differences here. So, the other thing that we ran into, and you'll recall I talked about those accelerometer and magnetometer sensors early on, and never really discussed them yet. Talk about them here, because what we found at one point was that we have predators that show up, and some of these predators are relatively clever. So this is a black oyster catcher, found all up and down the west coast. Let's see if this wants to play. Well, anyways, these oyster catchers walk around on these mussel beds right as the tide has started to drop or the tide is about to come in and has been splashing them. And they will start looking for not only little limpets to eat, but they will start looking for mussels that they can jab their beak into and eat. So here is a little bit, a short sort of time lapse of this plate, again, on the high shore. We're going to look at it over just the course of a few minutes, so we're taking samples, in this case, every couple of seconds. And what we're going to graph are the acceleration, how many G's of acceleration one of these mussels is experiencing. And then also the pink line is going to be how open or closed it is. So as we start this, the animal is closed, our pink line is very low, there's no acceleration. And then a few minutes into this, suddenly we see this massive amount of acceleration, and the shell is suddenly wide open. And notice at the end of our time lapse who's standing on top of our little bed of mussels. So this was very lucky. We just happened to catch this because it's happened 3 different times during the course of this experiment that we went out there and suddenly one of our mussels was wide open, completely cleaned out, and we went back and looked at the data and saw this same sort of pattern of a mussel that was nice, tight, closed, all of a sudden is wide open and has been whacked around like crazy. When you reconstruct what that looks like, this is a little sort of computer animation of the acceleration and position data. All of a sudden, this mussel looks like it's in a hurricane. It's just being whacked around all over the place. And you notice that the shells are sort of opening up in this reconstruction. Again, that's based on the data from this one particular mussel. And so this is an oyster catcher getting its beak in there initially and just start jabbing at it and then getting in there enough to move it around and rip out all of the guts of that mussel and make a nice meal. So these oyster catchers were super excited. We kept putting these little smorgasbord of mussels out for them and going, hey, thanks. So then transition to talking about stuff that we're doing down here in Southern California. In particular, looking at how mussels and oysters are faring in our local estuaries and bays. We're sitting here in the watershed that eventually feeds down into the Escondido Lagoon. If we go down one watershed into the sort of Poway area, that eventually feeds out into Los Penasquitos, which is where this picture is taken. And we'll talk a little bit about data from Los Penasquitos Lagoon, and then also down in the Tijuana River in San Diego Bay. So I'll introduce one other sensor that we figured out we can glue to these animals. In this case, it's a heart rate sensor. So if you have a Fitbit or an Apple Watch that tells you your pulse, it's doing that by shining a little LED into your skin and actually seeing the blood vessels change shape. And it's reflecting light back from that LED and picking up the change in that shape. So this is the same kind of sensor. This little chip here has a little LED, shines a red light in this case through the shell, and then picks up the reflected light coming back, and we aim it at where we think the heart of the animal is. So a mussel or an oyster has a heart just like us. And so as it sits there and pumps, we get this nice little trace. We get a pulse trace of what these animals are doing. And so you can imagine that much like us, as heart rate changes, that is often an indicator for all kinds of physiological changes. And so we're interested in how these animals are changing heart rate as things like temperature changes, or as they're opening or closing, or as they're being exposed to fresh water instead of seawater. So we're gluing these kind of sensors onto these animals, along with those gaping, shell gaping sensors, and tracking what happens out in the field. So here is a oyster cut open. You can see the little heart actually beating there. So that's what we're aiming our sensor at and actually getting that signal off. And we can do this, thankfully, without actually having to cut the animal open at all. We can just glue it onto the outside of the shell, confirm that we're seeing a reasonable trace, and then put that animal out in the field and have it live for months at a time like that. So I'll talk a little bit about a project we did first in Newport Bay, where a group of folks have been doing restoration of oyster beds. So Newport Bay is obviously a really heavily human-impacted bay, as you can see by the photo, the aerial photo. But it used to have a lot of native oyster beds all through the bay. And a lot of the development of the marinas and so forth sort of paved over or other dredged or otherwise removed those oyster beds. So back in 2016, there was a group that started actually putting out big piles of dead oyster shells. So actually shells that they were getting from seafood restaurants. And these piles that you see in the picture here are just a whole bunch of shell wrap. And they put them out in these coconut core bags to sort of keep them together initially, and then those break down naturally, and you're left with these lumpy piles. But the fact that there is now this hard shell substrate out there encourages native oysters to wash in as larvae and actually settle. Because what was here previously, or what's here in the rest of the lower and upper portion, is just sort of mud with the occasional rock in it. And oysters will not settle on mud. They need something that feels steady that they can anchor themselves to. And oyster shell is an excellent sort of indicator of, oh, this is a relatively safe place. I should settle down and glue myself with the rock. So we started putting sensors on native oysters that we were finding in Newport Bay, and then taking those animals and placing them in these restored oyster mounds, also placing them in areas where eelgrass, the green stuff, has grown up, and then also putting them out in just the bare mud that constitutes a lot of Newport Bay. So this is one of my master students, Lauren, who spent a lot of time gluing individual chips onto boards and soldering them, and then putting them in our little plastic PVC plumbing pipe housings that we could waterproof with a little rechargeable battery, and gluing our sensors onto these native oysters, and then all of that would go and get stuck in the mud, basically. So this is a photo of the oyster sticking up on the mud surface. The rest of that PVC pipe is shoved down into the mud and hidden there. And so over the course of basically a year, she was able to measure when these animals were opening and closing, and also what their heart rate was, along with their body temperature over the course of high tides and low tides, and rainy season and dry season and so forth. So she did this in those restored oyster beds, in this eelgrass, and then in the mud as well, and was looking for whether there were differences in the way these oysters were behaving or responding to their environment, depending on the type of habitat that they were in. With the thought being that, for instance, animals sitting surrounded by mud might actually get inundated by sediment more, because there's a bunch of sediment around. And so if it's wavy, they might just be getting a whole bunch of sediments in the water instead of nice plankton that they can filter and eat. Whereas animals sitting on oyster bed or perhaps surrounded by eelgrass might not get as much of that sedimentation. We might actually see differences in terms of how long they spend with their shells open, how much growth we see over the long term, etc. One of the biggest sort of signals that we see, not surprisingly I guess, is that rain and fresh water washing down from the watershed through these estuaries causes big changes in what the animals are doing. So the top graph here, the blue bars, are the amount of rain on each of the days in the area in the watershed immediately around Newport Bay. And then the black line down below is the group sort of average amount that they were open. So 100% open down to 0% being fully closed. And what you see is prior to rain events, everybody's basically spending most of their submerged time fully open. Once that rain hits and you get this fresh water washing through, sort of pushing this salty water out, that average in terms of how much time they're spending open drops. Big rainy days in particular are followed by often multiple days of these animals staying really tightly closed because all they're being bathed in is fresh water. And that change in the salt content of the water from ocean to rainwater is really quite physiologically stressful for these animals. So they basically clam up and try to avoid it. Just isolate yourself off from the world, wait a few days, reopen, taste the water, say, oh, it's salty again. I guess I'll reopen and I can start doing my normal routine. But this was the winter of 22-23, which, like the winter of 23-24, has been relatively rainy. And that was a period, you know, back in March of 23, where we just got a rain event every week for weeks on end. And so these poor oysters would just sort of have to spend 2 or 3 days of each week, more or less closed up the whole time, holding their breath, waiting for that fresh water to go away. So we would see these sort of patterns where in sunny weeks where it wasn't raining, most of these animals on average would be open most of the time. Rainy weeks, that average would shift down, and then the week following, when it was sunny again, they would open back up. And so over our entire year-long data set, the most stark changes were really related to that water coming down through the watershed and washing through the estuary. This is a whole lot of data, but I'm mostly showing it to you because we have heart rate data here as well, along with the rainfall in the orange bars there. So in periods where we had rainfall events, we tended to see the gate data drop off, get very low because animals were closing. We also tended to see the average heart rate drop a little bit as well. So what seems to happen is when these animals close up, I've said they're sort of holding their breath. They're not getting new oxygenated water flowing through their bodies. And so they are essentially sort of going anaerobic. They're using up whatever oxygen is trapped within the shells, and they're having to down-regulate their metabolism as a result. So they literally slow their heart rate down because there's no new oxygenated water to pump around, no oxygenated blood to pump around. So why waste the energy pumping your heart at a normal rate? So they slow down when they're closed up, and then once they reopen and they've got good oxygenated water, they bump that heart rate way up, sort of like recovering from an exercise session, or huffing and puffing as we do that. When the heart rate has been high, they sort of do the same thing, trying to re-oxygenate those tissues and get themselves back to a sort of normal level. Another version of this that we've been doing is using Pacific oysters. I guess I should say on the previous slides I was talking about the native oyster, which is fairly rare along most of the west coast of the U.S. these days. We do find it in the local estuaries occasionally, but most of the oysters that we're finding, you know, in San Diego Harbor or in some of the local estuaries are actually the introduced Pacific oyster that came over from Asia, was brought over from Asia, I should say. And the Pacific oyster is the one that you also get served in restaurants, the seafood restaurants. That's the one you're eating. And so we're looking at how those populations of those introduced Pacific oysters are faring in our local estuaries. And so I'll talk a little bit, as I said, about Los Penasquitos Lagoon. We'll talk a bit about what's happening in San Diego Bay and the Tijuana River estuary as well. These are all sites where we have these animals put out these sensors for months at a time to sort of gauge how they're doing in the field. Generally, the way we do this is we put sensors on a batch of oysters like this, running back to that box, and then we set it all up on a floating mooring that we can drop in the middle of the channel in the estuary. So these animals then aren't actually coming out of the water at all. They're not true intertidal animals anymore. This is like if they were growing on a floating dock and we were just always in the water as the tide went up and down. So we have the same thing here, except we're using a boogie board as our float. We have a group of animals up at the surface, just on the bottom of that boogie board, so they're really tasting those surface waters. And we have a group of animals down near our anchor, sitting in those deeper waters. And we go ahead and drop all that in the channel. here, in this case, in Los Penasquitos Lagoon, and let it run for months at a time and record those data. We also do this with mussels in some cases. So we'll start in Los Penasquitos Lagoon, and I'll show you some data. In this case, we're looking at opening and closing data again, so we're going to gate it. So each red line here represents data from one animal, so we have, what is it, five animals here. And then up at the top, we again have rainfall data. So these animals, in this case, the red lines, these are all animals that were up at the surface. And if you recall from science class, fresh water has a lower density than salt water. And so fresh water, when it washes through the estuary, tends to wash through in a lens on the surface of the estuary. It doesn't fully mix all the way down to the bottom. And so these animals that are hanging off our boogie board right at the surface, when the rainfall comes through, they're basically bathed in that fresh water. And what you see is those red lines go to zero and stay at zero. Each of these little dash lines here represents a full day. So these animals basically closed up for 4 days, holding their breath for 4 days. And then were able to reopen, and then they had another rain event, and they had to close for another 3 days, and so on. So we're looking here over the course from November through April, so multiple months worth of data. And when we had these large rain events, we would see, again, day after day of these animals closing down. So this isn't because of necessarily any sort of pollutants washing through, although that's always a possibility. We think this really is just being driven by the fact that it's that fresh rainwater, and they hate that fresh rainwater. The green lines here represent the animals that were down at the bottom, deeper in the channel, and so staying away from that freshwater lens. So the same big rain events where we saw all the red lines go to zero for multiple days, the animals that are literally 4 feet deeper in the channel, you basically see these little brief closures of a couple of hours where at low tide, perhaps there was fresh water getting close to them and lowering the salinity, and so they closed up. But they are not dealing with multiple day, 3, 4, 5 day closure periods. So these animals that are deeper in the water are really, really living a nicer life just because they don't have that stressful fresh water coming through. I'll show you then also data from the Tijuana River estuary. And if you know, if you read paper, you hear about the constant issue of sewage flowing through the Tijuana River estuary, going out onto the Imperial Beach coast, washing up to Coronado, causing all sorts of beach closures. The Tijuana River estuary does have lots of animals living in it. A lot of them die off every once in a while when things get bad, but there are oysters living there, there are mussels living there, and there are lots of other critters living there. And so we've gone out and put moorings in Tijuana River estuary as well to look at what happens in that way. And so what I'm showing you here is a whole lot of different graphs, but again, at the top is our rainfall data, so I'm going to show you data from one rainfall period. We're just looking at a couple of days worth of data here. This second to bottom graph is our gate opening, so this is data from a bunch of oysters, whether they're open, high values, or closed, low values. And then the very bottom graph is heart rate for these animals. And so these heart rate data are from the same animals that we're measuring the opening and closing. And we're going to just sort of zoom in here on a couple of days that I've highlighted in the blue boxes here. And so in this graph, we're zooming in even further. Those blue boxes represent the same events. And what I'll point out here is in the Tijuana River estuary, we had these rain events that caused big group closures. Everybody basically went to zero here, except for a little sort of testing period during a slightly higher tide. When they closed, we see those heart rates drop and basically cut in half. So a normal heart rate seems to be around 12 to 15 beats per minute. When these animals are closed up and holding their breath, they will drop down to 5 or 4 beats a minute. And actually, we can't, the way we're sampling, we can't reliably say that they're going to be lower than that. They may well be going lower than that. We just can't pick it up the way we're sampling. So I don't know that they necessarily stop their hearts, but they definitely get really, really slow. 1 or 2 or 3 beats a minute is awfully slow. And then basically, as we get high tides coming back in, that is salt water pushing back into the estuary, pushing the fresh water sort of up to the surface and holding it off. And so these animals will very quickly reopen, start pumping water, presumably start breathing, potentially feeding. And then once the tide drops again and the fresh water sort of takes back over and flows out, we see those animals all close down again, their heart rates drop again, and they sort of go through this cycle of having to close up versus reopen depending on what that salinity signal is doing. Now the other thing that potentially happens, as I mentioned, is sewage going through. And so we have these sensors out right now in Tijuana Estuary. And what we're obviously we're coming out of the rainy season right now. The interesting thing will be as we go into the dry season when there's less freshwater influence, or let's say less influence of rain events. Do we see any sort of these sorts of closure signals that might be linked to known sewage overflow issues coming through the Tijuana Estuary? So that's the sort of ongoing question for us at this point. The other thing we're doing is actually putting these on the internet. So at this point, you can get, like a $100 data logger, call it $200, that we can plug our sensors into, and we'll have a little cell modem on it, so you can put a little data plan card in it, and it will phone home to AT&T or whoever, and we can broadcast in this case, just the shell opening and closing data. So these graphs on the right here are data from the website. So when you see low values, again, we're closed up. When you see high values, we're open. And so over the course of about 3 days, you see this signal of open for a period of time, closed for a period of time, and so on. And you tend to see some synchrony there in terms of what in this case, 4 animals that are all living right next to each other, some synchrony in terms of when they're opening and when they're closing. But again, it's going to be linked to the tide cycle and also what the salinity of the water is. So this is, again, something that we've got currently running in the Tijuana Estuary, actually at that site, so that we can sort of keep an eye on things. It's also nice because I can sit at home and log in and see, are my sensors still working, as opposed to having to go out there repeatedly and check and see if things are alive or not, whether the animals are alive, but also whether the electronics are alive. This at least gives me a picture of, yes, the electronics seem to be alive. And then I want to finish off in this last section talking about another project where we're sort of looking at the effects of this idea about greening gray infrastructure. And so this is a project funded by Sea Grant and done in concert with the Port of San Diego in the San Diego Bay. And gray infrastructure in this case is referring to all of those human-built structures along the coastline, so all the concrete that's been poured along the shores of San Diego Bay. So I've got it here on the left, a drawing map of what San Diego Bay looked like around 1850, and then the Google Earth view of what it looks like currently. And there's, what I want to start with is the fact that San Diego Bay, when European settlers started showing up, was a whole lot of mud flats. It was not a bunch of seawalls and piers and so forth. A drawing from, this is 1876 here, looking onto, this is sort of Newtown, San Diego, Coronado across the bay, Point Loma off in the distance. What I want you to sort of take away here is that the shores of San Diego Bay sort of sloped very gently down to the waterline because there was a whole bunch of mud flats all around Coronado, all around Newtown, New San Diego, San Diego Town, and so on. That was all sort of originally created by the fact that the San Diego River, and then down in southern San Diego, we have another river system flowing in that were carrying sediment into San Diego Bay. And apparently, the San Diego River would change its course over every couple of years back in those days. So sort of zooming in here, we've got the San Diego River drawn flowing into the area that is effectively sort of where the airport is currently. Up here, you can see a sliver of what here is labeled as False Bay, which we know is Mission Bay today. But occasionally, the river in big flooding years would switch directions and flow up into False Bay and stop flowing here. And then a couple of years later, it would switch and go back into San Diego Bay. And this was obviously a bit of a problem for the people who were trying to build their houses and roads and so forth. So eventually, the river was channelized and sent into sort of the south of the Mission Bay as we see it now. But what it was doing in the old days was just dumping sediment out onto the shores of San Diego Bay. So San Diego Bay was a big mudflat. It looks different now, right? There's not a whole lot of mudflat left in San Diego Bay. You'll also notice that, for instance, parts of the Coronado have been filled in as well. And we've got, these structures like Shelter Island and Harbor Island. So Shelter Island was sort of at the terminus of where the San Diego River tended to empty out. So there was a sandbar originally that was sort of in this area along Shelter Island that in the 1930s they started dredging and sort of building up to turn it into more of a permanent structure. And then in the 1950s they actually connected it to the mainland. And then Harbor Island here was built from dredge materials coming out of the shipping channel in 1961. And so it's entirely man-made. There was no sort of sandbar there originally. And then the old boat channel here that runs along Liberty Station, that was sort of the continuation of where the river used to occasionally flow through. So I'm going to zoom in a little bit on Harbor Island in particular. And so what you get along Harbor Island is a whole bunch of rocky riprap. So this is stuff that was quarried elsewhere, trucked down to the shoreline and dumped along the edge of Harbor Island to create some armory along that shore, so that as boat weights would wash up on shore, it wouldn't just erode the dredged spoils that were building the rest of the island. So in sort of harbors all over the world, this sort of armoring of the coast is necessary to basically keep the coast where it needs to be, right? But it's not necessarily like the most hospitable habitat for critters. So this is a photo of the rock riprap, that it's called, along Harbor Island here. And what you see mostly is you see some oysters. Those are Pacific oysters, the introduced ones. There are some barnacles. And then if you get down in there, there might be the occasional mussel, the occasional little sort of snail or isopod crawling around. There's not a whole lot of stuff growing here a lot of the year. The other thing, though, you can't really see is there's some weird-looking structures in two spots. And so I'm going to sort of zoom in on those a little bit. And what those are these cast concrete blocks. And this is the part about greening gray infrastructure. So back in 2021, the Port of San Diego set up a pilot project to actually tear out some of the original rock riprap here and replace it with these cast concrete blocks. And these are about 3 feet wide. And they all have this little pool built into the middle of them. And so the idea is to basically still have armoring on the shoreline there, but have it be a structure that might actually support more growth of organisms. So this is a photo from the actual, initially scraping off some of the riprap. They have excavators and a barge that's been sort of scraping the old riprap away. And then they've got all their concrete blocks set up on the barge here, and the big crane can sort of swing them into place. And they're all positioned sort of very carefully next to each other and oriented so that most of them will have a pool, that tide pool in the middle. The ones very low on the shore that are low enough that they don't really need a tide pool, they're actually sort of oriented to turn that pool into a little bit of cave, a little overhang that might be, again, habitat for critters. And so we're interested in comparing what's growing on these new green infrastructure blocks versus the original riprap. And so that consists of going out there and getting down on our hands and knees and actually counting what's growing. So we've got various graduate students that have been doing these surveys for the last year and a half or so. And so we look at the original riprap along a sort of horizontal transect there and count everything that we can. All of the green algae, red algae, and brown algae that are growing on those rocks, along with all of the invertebrate animals that are found there as well. And the thing about the riprap here is that it really doesn't have any tide pools. It doesn't retain any water. Whereas those new blocks, they purposely retain a whole lot of water. So we're doing our surveys here. When you get to one of those blocks, to one of those tide pools, in this picture it's now about 3.5 years since it was placed, and it was bare concrete when it was placed. What you see is a whole lot of stuff growing
Stuff that in a lot of cases you will not, either you won't find that species at all, or you will barely find any of those individuals on the riprap. But when you provide them with a tide pool like this, you get a whole bunch of algae growing in there, you get a whole bunch of different species of algae growing in there, especially species that can't survive out on the hot, dry portions of the rocks. And then you get little fish settling in there, you get all kinds of little mobile critters. This is a photo of a sea slug navanax that we find those occasionally in the pools, crawling around within there looking for stuff to eat. So we get a pretty wide variety of critters growing, settling and growing in these blocks. The other thing we're interested in is what the thermal environment, the temperature environment is like on these cast concrete blocks versus the original riprap. So we're actually monitoring temperatures in the tide pools, out on the exposed portion of the concrete, out on the original rock riprap as well, comparing sort of how stressful those different microhabitats might be or not stressful in the tide pool. And we're also doing that by actually building little fake animals and putting them out there with temperature sensors in them. So in this case, we've got little cast silicone crabs, we've got little limpet shells filled with silicone with temperature sensors, and that's what each of these wires is. And so we'll put those out at low tide on nice, hot, sunny days, and put them on the sun-exposed portion of the rocks, put them on the crevices, and see sort of how stressful or not stressful those various portions are. And look at both mobile critters that might have the ability to sort of choose where they want to be, and also what we call sessile organisms that can't get up and move, and sort of where they might survive or not survive. I won't show you a whole bunch of data here, but just to sort of show you that when we do our surveys at this point, on the old rock riprap, we find 24 different species of algae and invertebrate animals. In these new concrete blocks, that number goes up to 35, and those species tend to mostly be associated with those tide pool habitats, which just are not present on the rock riprap. So we find a couple of different species of seaweeds that we wouldn't expect to find on bare rock, but they will grow in those tide pools. And then we find a bunch of different small invertebrate animals associated with those tide pools as well, that like to live among all of that lush algae growing within those tide pools. So I'll wrap it up there. Acknowledge a whole bunch of people that have been involved in this and the various different projects, a whole bunch of funding agencies that have helped support this work as well. And I'd be happy to take questions if you have.
Yeah, I've got a couple questions. So if they're closed and they're being bathed by seawater or they're not, and they open up when they somehow sense the seawater, how do they sense that there's?
So what we've decided must be happening is they're just sort of barely cracking that shell open, just a fraction of a millimeter, not much at all, letting some exchange happen there, and then they've got cells that have salinity, effectively salinity sensors, sodium chloride sensors, like our taste buds essentially, and they're tasting that water. Going, is it fresh or is it salty? If it's salty, then maybe I want to go ahead and open up.
So how do they do that on some regular basis? So if the tide comes in, the tide goes out.
Yeah. So we don't see a really regular pattern. What we see is that when the tide has been high for a while, various individuals at different times will sort of crack open a little bit, and then usually they just sort of keep going because it tastes fine. It's on those periods when it's just rained and we're getting the fresh water flushing through, where we might see tiny little openings and then closures, and then they may stay closed for a couple of hours still after that. So it's not like every 10 minutes they're testing again and again. It is on that hour base, or multiple hours time scale that they go in and test.
Assuming during rainfall, fresh water's coming down, so into the Tijuana River coming out, would increased heart rate then indicate pollution? And can we use that data to say, hey, look at this is one of the problems that's going on. That's sewage water coming in during rain events is creating poison, in our.
So what I expect that we will see is that actually it will be a signal very, very similar to just the let's say pure freshwater signal. In that if they start tasting stuff in that water that they don't want to taste, that is indicative of pollutants, they will close up, clam up, seal themselves off, and do that again for maybe hours or a day or two, and reopen and test. And so during that time period when they are closed, we expect those heart rates to drop. We expect them to stay low to just save energy, because they are literally holding their breath during that period.
So given that data, then just reinforce
Yeah, so where we expect to then see that signal is we have to couple it with the water quality data that's also being generated. So we know it hasn't rained recently, so we know there's not some major flushing event happening. But we're seeing these animals all close at the same time and stay closed for long periods of time. That's potentially a signal that they're tasting something in that water that they do not like. And so that's where we probably expect to see that. So the heart rate data, in the long run, the heart rate data probably aren't going to be that important a metric for that sort of water quality sensing part of it. We can probably just boil it down to whether they're open or closed. To me, it's interesting from a sort of physiological standpoint, the sort of, let's call it more pure science questions about what that heart rate is doing. But from a water quality monitoring standpoint, I think just the opening and closing data is going to tell us 95% of what we need to know.
Right. My other question is, we're hearing a lot about coral reefs bleaching out. Are you finding that along California coast? And because of temperature change?
So in the systems that I study, they are traditionally exposed to really radical fluctuations in temperature just on a day-to-day or even seasonal basis. And so we don't see those sorts of mass sort of die-off events that they observe on coral reefs. Now, coral reefs have the very stark and visually apparent signal that, everything goes white because all of the photosynthetic rhinoflagellates have been released out of the corals, and you're just seeing the calcium carbonate skeletons. On our coast, we don't have proper corals like that. But we've got, when algae gets stressed, they can actually end up bleaching out, and then they generally just flat out die at that point. But we don't see widespread bleaching events in algae like that. I think partly because our algae are just hardier than corals are. Now, corals are living within this very narrow range of temperature fluctuations that they will ever see over their lives. It gets warmer in the summer, cooler in the winter, but we're talking about 3 or 4 or 5 degrees, not 20 degrees.
Are the temperatures more consistent along California coast than, let's say, Florida?
They're more consistently different, I guess is what I would say. So we get the cold winters, we get the hot summers, and the animals that live here and flourish here have been dealing with that over evolutionary time. So they're just sort of built for it.
Right.
Is the local population of the animals you're talking about, is it hardy?
They're
thriving?
I mean, the oysters and the mussels that we're looking at, I mean, when the ones that we're putting out in places like Tijuana, we walked out there first and grabbed them from the shore on the Tijuana estuary. So they've been growing there since they were babies and putting up with these conditions all the way through adulthood and presumably reproducing. So yeah, they are pretty hardy. The place where we see die-offs in estuaries locally is when the mouth of the estuary is closed off by waves piling up sand on the beach. And so Los Penasquitos Lagoon has that happen pretty much every year. Tijuana has it happen every couple of years, and it's actually, I think it's closed since 2016 right now at this point. Whereas Los Penasquitos will close 2 or 3 times over the winter. And it's done so this winter. And so when the berm forms at the mouth of the estuary, the water's not flowing out, and we're not getting good ocean water coming in. And so we just get this sort of continual drainage of fresh water into that estuary, all the runoff from people's irrigation. It's not that we need a rain event to make it bad necessarily, just sort of standard amounts of runoff will build up in that trap in that estuary, turn the water fresh. Temperatures tend to go high. The oxygen tends to get used up because it's not being flushed out and mixed. And so that's when we tend to see die-off events. So in 2021, I think it was, winter of 21, at Los Penasquitos Lagoon, there was a sandbar across the mouth. It stayed closed. Actually, no, it was 2020 because we had that huge bioluminescence event that lasted for like a month and a half off of scripts and so forth. that water with the algae that were creating the bioluminescence, some of that got trapped in Los Penasquitos Lagoon just as the mouth was closed off. And so there's this nasty, red, soupy water there. The animals were dying, the bacteria were breaking down. In the process, they were using only oxygen in the water, so the oxygen went to zero within the lagoon. And we had animals dying like crazy as a result, because there was no oxygen in the water. It lasted for multiple days like that, and a lot of animals are not quite as hardy as the oysters and mussels, who could put up with a couple of days. But you know, your little snails crawling around, or some of the worms living in the mud, can't necessarily deal with multiple days like that, so they just up and die. And particularly fish are particularly susceptible to that. If they can't get out of the lagoon, get out into the ocean, they're just out of luck.
So the temperature data that you showed early on, do they generate their own body heat, or are they absorbing it from the sun?
So they're all, the vast majority of that heat is coming from the sun. These are cold-blooded organisms, and their metabolic heat production is there, but it's very, very tiny. So it's not on the scale of warm-blooded critters like us. So when we sort of build models for estimating temperature in these things, we ignore metabolic heat production entirely. We just say this animal is not making any heat on its own, even though it's probably making just a fraction of a degree or something like that, but it's minuscule.
So the end game of this research, are you guys hoping or looking for funding, or who would be kind of greening the rest of that? Using that like artificial?
Yeah, so the Port of San Diego is interested in this because, for one, the riprap in some of these places is now from the 1960s. It's old. It's going to need to be maintained, and so they're sort of doing this pilot project to get at the question of, is it worth it to invest a little bit more money to do these precast concrete blocks? And then there's definitely, it's not just tossing stuff over the side of the dredge. It's got to place them just right with a crane. But is it worth it from the standpoint of still continuing to armor the shoreline, which is their primary worry? They don't want Harbor Island washing into the bay. But can we also get some of what they're calling sort of blue carbon, blue economy benefits from that by having more critters living there and pulling the CO2 out of the atmosphere, pulling CO2 out of the water, sort of filtering water, cleaning phytoplankton, and all that sort of stuff. So that's sort of their thought, is that there's the dual benefit in this case.
Is there any con besides a little more than
There's the CO2 production from actually casting the concrete. So it's not like it's a net zero necessarily process in that regard. Cost is the primary driver, and I mean it's as I sort of alluded to there at the beginning, ultimately all of this is not getting us back to what the natural state of San Diego Bay was. The natural state was a whole bunch of mud, and we're not going back there anytime soon. So this is the sort of compromise situation where the economic and recreational components of the port's mission are upheld, but we get some environmental benefit out of it, hopefully, as well. All right. Well, if that's all the questions, thank you so much for